sort of have two hats. One of them is more of a neuroscience, kind of traditional scientist hat. The other one is working on open source tools and sort of like new models for how science can be done collaboratively and in sort of an open source kind of way. So what I want to talk about today is a short overview of the world of visualization in the human brain. And I'm going to stick my face closer to the microphone so that you guys can understand me. So this is going to be a little bit of a sort of background about what brain visualization is, what it has been, and maybe what it's going to be. And then I'm going to go into a few tools specifically in the Python ecosystem that we have for visualizing brain activity and sort of making it easier to adopt particular kinds of analyses. So the problem with neuroscience, and I know this doesn't sound like a problem, but it really is that it's really noisy, it's really complicated, and it's really sexy. And this is sort of this perfect storm of factors that result in, shall we say, like dubious science getting published in really high-profile journals and things like this. And I think that for this sort of reason, it's really important to have tools that let you ask really powerful questions about the brain, but that don't encourage you to sort of do the wrong kinds of things with your analyses. So fundamentally, neuroscience cares about two different things. It cares about things that happen in the brain, and it cares about things that happen in the world. And 90% of especially cognitive neuroscience is basically just trying to relate these two things to one another. Now we have lots of ways that we can record information about the world. You know, you can use a microphone or a video camera or information about when things happen. But how can we actually see what's happening in the brain? Brains are these sort of impenetrable, really complicated, difficult to understand things. So more than 100 years ago, a guy named Santiago Ramon y Cajal basically became interested in this question, and his solution, because this was the 1800s, was to just look at brains at very, very close views and literally just draw them with a pencil and a paper, which sort of, I imagine this is like the first open source tool set, pen and paper. And what you can see here is basically his rendering of what's basically a slice of your cortex. And the reason I show this is for two reasons. One is that visualizing the brain for whatever reason has always been important to neuroscientists. Basically, as long as we've been interested in the brain, people have tried to visualize it. Two is that, as you can see here, there's a particular structure that exists to these neurons. This is basically a very tiny sheet that's right on the surface of your brain. And if you may notice, there's a certain orientation of these neurons. They seem to go sort of from bottom to top. And that turns out to be important a little bit later on. So unfortunately, we don't want to study the brain by just drawing things with a pen and a paper, because this takes a really long time. Over the next 80 years or so, our ability to visualize the brain really improved. At some point, some scientists realized that we can basically harvest a protein that existed in jellyfish. And this is a protein that when you expose it to a particular kind of light, it will fluoresce. It will actually generate light. What people were able to do then is express this protein in neurons. And you get really nice single neuron views like this. And again, this is sort of happening automatically. This is a neuron that when you shoot a photon at it of a particular type, it's going to generate this really beautiful image for you here. This is called GFP, or green fluorescent protein. So what scientists have done is they've sort of improved their ability to visualize the brain with this sort of GFP approach. They have lots of other colors that you can use. You can express them in different kinds of neurons, for example, to see which neurons are of a particular type, or you can kind of manipulate them experimentally, whatever you'd like to. And there are a lot of tools that exist to do things like cell segmentation, image segmentation. You might want to pull out all of the red cells here. And a lot of neuroscience still is just like counting these things, like looking through here and saying how many cells of a particular type do I see in this particular sample. But this is sort of a very static view of the brain. And especially the human brain, what we really care about is the sort of dynamic, ever-changing nature of the brain. So in order to understand that, we need time. So in order to understand how time operates in the brain, I think it's important to understand what a brain is actually doing in a dynamic sense. Now, some of you may know that brains are essentially made up of these little things right here. These are called neurons. On the left are what you can think of as a bunch of little receptors that take in electrical signals. On the right is a sort of transmitter that transmits an electrical signal. I should note that by electrical, I mean chemically electric, but I'm not going to go into that detail here. Essentially what happens is these little neurons are everywhere inside of your brain, And when they take in information from their environment, that information gets collected there, that little green dot, it's called the cell body. And if enough excitation comes into the cell, it fires what's called an action potential. And this basically means that it shoots an electrical signal from that little green circle in the middle down the axon, and then it ultimately results at some chemicals being released there at the far end on the right. The reason that I show this is because when you have an electrical charge that moves from one side of a neuron to the other, it generates an electrical field. A very, very tiny one, but it is a field nonetheless. And again, that turns out to be really important. So we have space and we have time. What does this kind of thing look like in action? So I'm going to cheat and I'm going to show you a movie that's not from humans, but I'm going to show it to you because it's really cool. This is a zebrafish. You may wonder why we care about zebrafishes, since they are obviously very far from humans. Well, as you may notice, zebrafish, when they're really, really young, are transparent. And what that means is that you can cause their cells to express light when they're active, and you can basically see their entire nervous system all at once. So as an example here, you can see the zebrafish from the top down on the left. On the bottom right, you see the entire zebrafish. You can see its eyes in the top right, and basically what you'll see is that there's a little tiny, what's called a paramecium swimming in front of it. And you can get an idea for how this zebrafish's brain is responding to that little paramecium. So you can kind of see, the little guys, every now and then it swims in front of it, and the zebrafish is like, whoa, I want to eat that, but it can't because it's stuck in gel, which kind of sucks. But anyway, you can see its brain activity firing, trying to generate those action patterns, taking in its visual perception, trying to translate it into a decision, and then ultimately some sort of motor activity. And the reason that I show this is because it's really complicated. There's all kinds of crazy stuff going on. And so that takes me to what I like to call the trifecta of neuroscience. These are sort of the three best things that we want to have as we ask questions about the brain. The first is we want to know when things happen in time. Another way of saying that is we want temporal resolution. The second thing is we want to know how finely things differ in space. If there are two cells that are right next to each other, we want to be able to say that cell A and cell B are doing something differently. And then the final thing is that we want to record from lots of things all at one time. So for example, if I take an electrode and I stick it into a mouse's brain, say, I can record from 1 or 2 or 3 neurons, but that's 1 or 2 or 3 out of many, many millions of neurons. So how can I really make a statement about the entire brain if I'm only recording from a very small subset? And the challenge of neuroscience is that we don't really have a method yet that accomplishes all three of these things at the same time. Okay, so we can just take this technique, we can get humans, we can genetically express them so that they're translucent when they're babies, we can stick them under a microscope and look at their brain activity. Unfortunately, no, that's definitely not possible because A, it's scientifically very difficult, and B, you'd be in jail pretty quickly if you tried to do something like that. And the reason that I sort of make this point is that this is where neuroimaging becomes really important. I say the word neuroimage, and I'm going to sort of point it out specifically because I'm kind of interpreting image in the definition of a representation of something. You're not recording the thing that you really care about in cognitive or human neuroimaging. You're recording some projection of that thing. You're recording a representation of that thing. And so the big challenge in neuroscience, in cognitive neuroscience, is that since we can't visualize the brain in all of its complexity in the way that we just looked at that zebrafish, we basically have to try to understand what's going on in the brain by looking at signals like this. So this is an example of what's called EEG, which I'll get into in a second. But this is sort of what we can look at as scientists. And from that, we're trying to infer all of these things that are happening down at the cellular level, which it turns out is a really, really hard thing to do. So the two main techniques that we have in neuroscience asking questions from humans are called EEG and fMRI. So remember how I noted that in those Ramon y Cajal drawings, it seemed like the cells were sort of oriented in a particular direction. And remember also that when a single neuron fires, it generates an electrical field. So it generates what's called a dipole. So it turns out that if you get a whole lot of those neurons, and they're all firing at roughly the same time, and they're all oriented in roughly the same direction, then those little tiny electrical fields sum together, and they create a large electric field. So you get a whole bunch of neurons, they're firing like that. It's generating a pattern of electrical activity, and that activity propagates up to the surface of your head, up to your scalp. So what EEG is, or electroencephalography, it basically just means we're taking little tiny electrodes, we're putting them right on your scalp, we're recording the fluctuations in electrical activity that are right there, and we're trying to infer all the stuff that's happening deep inside of your brain just by looking at its sort of projection as it exists on top of your scalp. So as you can imagine, this is a really challenging thing to do because, for lack of a better way of describing it, a whole lot of stuff can change between deep inside of your brain and the signal that we ultimately record on your scalp. So if you look at old visualizations of this kind of thing, they're usually these very, very simplified summary statistics. This is what you would call a scalp topography map. It basically says if we put a whole bunch of these electrodes on top of your head, what does the amplitude of the signal look like as it's distributed across parts of your head? And a huge, huge part of neuroscience, endless debates at conferences, friendships ruined, relationships ruined, come from debating how patterns of activity deep in the brain can result in those patterns that we see up there. So that's EEG. The second big thing is called fMRI. And this is basically a recognition that whenever those cells fire there on the left, they require nutrients in order to stay alive. The idea is that if more cells are firing, they need more nutrients, and your brain has this sort of natural compensation effect where it tries to send more blood to regions that are more active. That's the idea anyway. So if you have all these blood vessels, they're active, maybe we can use some sort of technique to try and record this blood flow as a proxy for neurons that are kind of doing stuff in a particular region of the brain. Now, the way that we do that is very complicated and sort of beyond the scope of this conversation, so I'm just going to say that it's magic, and basically what it results in is some proxy for brain activity as it is related to blood flow in the brain. And in fMRI, you get a much better sort of spatial idea of what's going on inside the brain. So this is a really common visualization. You get these slices of the brain. One thing you can notice is since this is an MRI machine, you get a lot of anatomical information about what's going on. You'll get these summaries where you basically say like, okay, there's like condition A, condition B, condition C. We recorded the average activity for each condition on a bunch of different repetitions of, say, some stimulus, and we just want to know if there's, in general, a difference between them. You can also see this really cool, cutting-edge, three-dimensional visualization of the brain right here. I should say, this is all from like 1993, so it actually was pretty cool at the time. But the point is that here, we really lose information about how things change in time, but we gain information about how things change in space. However, the thing that really joins this, as well as the previous example with EEG, is that both of them are these very simplified views of what's going on in the brain. They're basically taking all of this complexity and they're reducing it down into just a couple of numbers because it's so difficult to make sense given the variability and given all of the sort of steps in between the initial brain activity in the neurons and the sensors that we're actually using to record them. So that kind of brings up this question, you know, in the age of the modern computer, how can we actually try to leverage this complexity or at least do less kind of reducing and removing of that variability? How can we try to sort of recognize it as a feature instead of a bug? And then sort of related to that is an extra question of how can we study the brain under more natural circumstances? So one of the problems if you have, you know, trial A and trial B is that that's not how the brain experiences the world. That's not how you have evolved and how you have grown to understand things. Events don't just happen in these nice little sequenced block style events. But if you want to present a more complicated situation to, say, a subject or to a person in an experiment, you also need more complicated ways to understand and visualize their brain activity. So people often show plots like this when it comes to scientific disciplines. You have like the neuroscientists on the left and you have the computational neuroscientists on the right. And all the neuroscientists ask the computational neuroscientists like how the hell they're supposed to analyze their data. My view, because I'm sort of in the open sourcey, maybe like stubborn scientific world is that while there may be neuroscientists and computational neuroscientists, they are all neuroscientists who use computation. And in my mind, in the future, anyone who's trying to be in a scientific discipline needs to know programming. They need to know an open source language. They need to be comfortable and flexible in how they use that language in order to ask their questions. So a lot of what I do is work on open source tools to try to sort of enable that for them. And so in the last part of the talk, I'm just going to demo a couple of tools that currently exist in the Python ecosystem. These are roughly split by the modality that we're recording from. So EEG on the left is a package called MNE Python, which is part of sort of a broader ecosystem, but I'm not going to get into all the details there. On the right, fMRI has had maybe a 6 or 7 year head start on electrophysiology because they've had these data problems maybe a little bit earlier in that field's history. So there's sort of an ecosystem of tools that exist for asking questions in fMRI. So what I'll do is just sort of talk about how each of these different things approaches both the analysis pipeline and also how it tries to incorporate visualization into the workflows that it tries to enable users to do. And for that, I'm going to attempt to do this live, and we'll hopefully not regret this horribly. Okay, so brain visualization in Python. The first thing that I want to talk about is MNE Python. I should say this is a project that I personally am a core contributor for, and as such, I think it's definitely the coolest and the best of all the things that I'm going to talk about here, although everything is pretty cool, I have to say. The goal of MNE is basically to try and minimize the amount of like really tiny little pain in the butt things that you have to do in order to ask questions about your data. So I'm just going to load in some raw data, and basically this is an experiment in which people were presented two kinds of stimuli, either a checkerboard pattern that they looked at or an auditory tone that they heard. They had a whole bunch of EEG sensors attached to their scalp, recording the electrical activity in their brain as they were looking at these different kinds of stimuli. And then the question is sort of how does the brain's processing of these different kinds of stimuli, auditory or visual, change the distribution of activity that you see on their scalp. So this is an idea of where these sensors are. You can kind of see the nose here at the top, the two ears here on the sides. Each of these little dots is a sensor, and that sensor is recording the electrical activity in their brain. So as I mentioned before, we can plot the raw data pretty easily with sort of simple tools like matplotlib. Here's one channel. Here's a whole bunch of channels over time. As you can see, this is roughly 8 seconds of data. And there's a whole lot of stuff going on in here. A lot of neuroscience basically says, like, I assume most of this stuff is noise, and I'm going to try to remove it. And we'll do a little bit about that here. But I think that, again, maybe sort of a future-facing approach is to try and see if there might be something interesting going on in those little tiny details as well. But either way, one of the things that MNE tries to really enforce is that it's really important to look at your raw data, and it tries to make that very easy to do. So I'm not going to do any interactive stuff here, but this is basically one of the sort of first things that you do anytime you get a new data set. You should be able to sort of scroll horizontally through different moments in time. You can scroll vertically through different kinds of channels here. I'm not going to do it because every time I try to do this interactively, I go way over time because I get too excited doing all the interactive stuff. But either way, this is what the raw data looks like right here. So as we mentioned before, our goal is to take this raw data and try to relate it to things that are happening in the world. And the simplest way to do this is to define event timings. Basically, when do particular events occur, and what does the brain activity look like around that event? So here I just have the indices of when a whole bunch of events are happening, and I can just take that same raw activity plot and show when each of these events happens. So it looks here like we sort of have like event 1, event 2, and then there's a space in event 1 and event 2, and there's a space. Notice it does have this kind of like blocked, maybe artificial sort of design. So what we can do is pretty easily say, okay, I want to take my raw data right here and these event times, and I want you to give me a window around each one of them so that I can look at sort of what's going on around the time that that event occurs. So basically what we need to do is reshape our data where it was initially of shape channels by time. Now it's of shape trials or epochs by channels by time. Again, we have a tiny little window around each one. And we can pretty easily generate another visualization that just tries to see something that might be going on in response to these tones or these images. Now, this is sort of the challenge with EEG. If you look at this, again, this is raw data, each individual trial. The green line is time equal to 0, so that's when the event starts. And each of these black dotted lines here are boundaries between events. So each column, 3, 1, 3, 1, 3, these are an individual event happening, and it's a window of activity around that event. If you look at this, I mean, I don't know. I don't really see anything obvious that's happening. And this is sort of a big challenge with EEG. The signal to noise is really, really bad. So what we want to do is we want to be able to average out that variability. The way to do this is quite easy. If I call average right here, it's just going to say average across the event's dimension. And so what I'll get back is something that is channels by time. It represents the average response of each channel to a particular event type. And if I plot that, now it starts to seem like there's something potentially going on here. So again, each one of these lines that I'm plotting is a different channel, aka a different electrode that's on that person's scalp. And you start to see some shape coming out of this. There seems to be some particular structure that is happening in response to each type of event. But it's really important to be able to incorporate different kinds of information into this visualization. So this is something that tells us a lot about how things change in time, but what about how things change in space? So for that, we can basically visualize the same thing, only as a two-dimensional plot. So now the x-axis is still time, the y-axis is channel, and the color represents the amplitude of each channel. And so instead of seeing them all superimposed, we can see that in some sense there's a structure here where channels that are sort of near one another are also showing very similar patterns of activity. So how can we sort of relate this to things that are going on in the brain? For that, MNE has a function called plot joint. And basically, what this does is it sort of follows this principle of trying to pack as much information as possible into a single visualization. And that all gets back to this question about how do I get both spatial resolution and temporal resolution and a sort of broad picture about what's going on in the brain. So what I'll do now is I'm going to break it down by two different event types, one being sound and one being visuals, and look at what seems to be going on in the brain. So as you can see here, I've now colored each electrode by its location on the scalp. In the top left there, I'm showing you where each of those electrodes are. At these various moments of interest, I'm showing you the distribution of activity that exists on the scalp. And so, for example, we can see that after a sound occurs, so sound is on the top, vision's on the bottom. After a sound occurs at about 100 seconds, there's this particular pattern of activity that you see that shows up. And this is very different from the pattern of activity you see when you look at a picture. And you would sort of expect this to be true because there are completely different regions of your brain that care about sound versus images, for example. So really, the point of this is that it's really important in terms of designing software to give your users flexibility and sort of say there are lots of different angles that they want to take on the same data. It's very complicated, it's very noisy, and so the challenge is basically to give them many different knobs that they can turn and many different ways that they can approach the same fundamental data set in order to understand something about what's going on. So the second thing that I want to mention is a way in which some of these tools are trying to incorporate the visualization and the sort of standard data analysis munging pipeline with the specific analysis that someone is going to end up doing. Because at the end of the day, we're doing all of this stuff to ask questions about the brain and hopefully say something interesting. So Scikitlearn and you probably all know what Scikitlearn is. It's a standard machine learning library in Python. Scikitlearn is basically a collection of functions that try to allow you to understand and ask questions about the brain, sort of keeping the Scikitlearn ecosystem and API in mind. So I'll just import Scikitlearn and give you an idea of what we're dealing with. The task in this case is a person that was sitting in an fMRI machine and they were looking at two different kinds of pictures. They were either pictures of cats or they were pictures of faces. I swear to God, this is the kind of experiment that people do in neuroscience all the time. So we have the brain activity in response to these two different kinds of objects. They're both visual. What do we think could be going on in the brain? Because these are really awesome open sourcey neuroscientists, their data exists freely on the internet, and so we can really quickly download it and take a look at it. So in this case, we no longer have channels by time as a data representation. We have X, Y, Z, and time. And that represents the fact that in fMRI, we have a much finer detail about where things exist in the brain. This is a volumetric representation of your brain. We can immediately plot this and get an idea for what this looks like. So this is the person's actual brain that this data set was recorded from. And we see we have all this information about anatomy, what's going on in these different parts of their head. This is what the actual activity looks like. So this is what's called the bold activity. It's the sort of fMRI thing that we record. And you can see that there's all kinds of stuff going on all the time. Now this is activity in response to a single trial. And what I'm not going to be able to show you is that if you look at the activity over time, it's much slower. And that's one of the drawbacks of fMRI. As opposed to EEG, which is moving very, very quickly in time, in fMRI, you're fundamentally measuring blood flow. And blood changes much more slowly than electricity does in the brain. Okay, so as you notice, we have this basically cube of information about what's going on, a cube around the person's head about what's going on inside their brain. But we really don't want that whole cube. We just want to select a subset of different parts of their brain that we might care about. And in order to do this, neuroscientists use what's called a masking technique. And this basically says, I want to define a region of the brain that I really care about. I want to extract that region. I want to just take those signals, do some analysis on those signals, and then sort of project it back onto the brain to figure out where those signals existed. And this is one of the ways in which NiLearn tries to sort of keep in mind Scikitlearn in its workflow. So I can plot what's called an ROI, or region of interest. This is basically what I mean by a mask. I mean, in this three-dimensional space, I want you to just give me the voxels that correspond to that person's brain. And people can use this in different kinds of ways. Sometimes they just try to extract the brain. Sometimes it's a particular region of their brain. But we're just going to use all the voxels only from that person's brain in this analysis. So they have a tool that's called a nifty masker. And basically this says, take a three-dimensional representation over time and basically transform it into a shape that I can shove into a Scikit-learn pipeline. Give me something that is samples by features. And it basically acts the same way that any kind of Scikit-learn transformer would. So now what I can do is just pull out the labels for images that I really care about for this kind of an analysis, and I'll fit just a standard support vector machine to basically say, do patterns in these voxels differentiate between faces and cats? So what's cool about this is that if I'm using support vector machines, I get a coefficient, one for each voxel, basically. And because I have this nifty masker and it kind of knows about how to transform things between space and between Scikit-learn sort of shapes, I can do the opposite of that. I'll say, give me the inverse transform of these coefficients. And when I do that, I get back this image. And this is basically the sort of standard neuroscience image that people use to make plots. And so using that, I can plot a statistical map that basically says, where in this person's brain did the brain activity differentiate between these two kinds of things? I can pretty easily set a threshold here to make sure that we know where the really important coefficients seem to be, or the really large ones. And again, this is sort of an easy way to like very quickly see where things might be going on in that person's brain. So the point of this is, it's really useful if you design your visualization tools and your analysis tools with other kinds of tools that exist in the ecosystem. And it makes it really easy for users to do lots of different kinds of things with the data. So one very final thing that I'm going to show here, and this can be pretty quick, is what I think is going to be sort of the future of visualization in neuroscience, and it's basically interactive and three-dimensional plotting. A lot of this is in conjunction with Jupyter, which is really cool in that it sort of leverages all the power of your browser and JavaScript and WebGL in order to do things that you wouldn't otherwise be able to do. I'm not going to go into a whole lot of explaining because this is basically like bleeding edge stuff. There's a decent chance that I'm going to crash my computer trying to do this. I was just working with some of the Jupyter team last night to get this working. But basically what we can do is define a bunch of geometry objects that allow me to visualize one of these brains within the browser itself. So here is that person's brain, and I can sort of look around it and look inside the different gyri and sulci and stuff like that. It's kind of cool. The other cool thing is that it is interactive. So if I take that original plot that shows that person's brain, I can really quickly say, like, show me all the colors, or change all the colors for all of these faces. And when I do that, I immediately get an update that shows me what's going on in the brain. So this is just some garbage stuff that I put together for this talk, but you could imagine doing something like this to visualize the dynamic patterns of activity that exists over time. And then I'm not going to actually show you this movie because it's pretty awesome. Actually, you know what? I will, but I'm going to walk off the stage as I do it. The last thing that I want to mention is another another package called PyCortex, and this is something that basically attempts to combine visualization with the sort of results of analyses themselves. And what it allows you to do is show the kinds of stimuli that you're presenting to the person along with the brain activity as it's changing. It's using Three.js under the hood in order to accomplish all of this stuff. And what it allows you to do is give these sort of complicated, changing, almost like demonstrations about what's going on inside the brain. And what's cool about this is that it allows you to start asking much more complicated things about what might be going on inside of that person's brain. It can sort of give users and give viewers a little bit more intuition about questions that they might ask about which regions of these brain are responding to, in this case, a natural stimuli here on the bottom. So the point of this is basically that we're getting closer and closer to observing the brain in its more kind of naturalistic context. So I'm going to really quickly load these things here. The main things that we've learned in terms of open source neuroscience analysis is embrace messiness by being able to show lots of views of the brain, to develop visualizations that keep other kinds of tools in mind, and then finally to incorporate interactivity in your analyses in order to sort of let people ask more complicated questions than they otherwise would. So thanks very much, and if you have any questions, I'll stick around today. I can take questions until I get kicked off the stage, which I think I am right now. I cannot take questions.